Yeah. Do I have some slides? Oh, there we go. Thanks. Okay. So, so today I'm going to talk about the role of the EM unit with respect to kind of projects in the country. And this is actually this is not my project. So when I'm presenting it, I don't know exactly what's going on with the project, but I'll try and present it the best way I can. Um, and I'll kind of explain why I'm presenting somebody else's project soon. So the Electron Microscope unit is on the campus. We're just behind this building over there. Well, a few buildings back, I guess. Um, the, the facility is quite nice. I mean, it's a nice building. We have nice offices, sample preparation, a data center. We've got a room that currently is occupied by material science microscope, but we'll just take that out as soon as we get the Titan class. Material scientists are laughing over there. Okay. And we have a range of different instruments. So these are instruments really for the scientific community, not only at UCT, but throughout the country. So it's a few different instruments, quite a large range, including this very interesting instrument which collects a Raman signal. So in other words, you can get clinical information about a sample. And then you can correlate that with its structure because it's a correlative microscope that goes between the scanning electron microscope and a Raman microscope, including TEM microscopes, EM sample preparation. Okay, so 7 members of staff, many of whom are here today. And this really means that this unit provide a lot of support. We can really do a lot. So just a few statistics about the unit. So about 3,000 microscope hours are provisioned annually. There are over 300 users. 80% of those users are from UCT, 6% from industry, 14% from other universities. Approximately, now this is kind of difficult to gauge. There are approximately 25 UCT graduations that have some kind of data from the electron microscope unit towards their graduations. Now, these are not necessarily people that are doing wholly microscope related projects, but there's some contribution. Around about 20 UCT publications per year. There've been a total of 42 EMDB depositions, 46 PDBs. And currently, we're doing around about 5 international data collections per year. So that's either at the ESRF or at Diamond Light Source at EBIC. Okay, so this is kind of what it looks like. So in other words, these over here are reconstructions that happened prior to 2018 when we really started doing lots of international data collections. The interesting thing about this, so the first reconstruction you see here is from Trevor. That was from 2003. And then over the years, various people did various reconstructions. Now, the first thing you'll notice is that there aren't too many of these structures because every single one of them was really tricky. So in many cases, these kind of reconstructions would take a person about a year to do because they would really have to go through this really slow process. And the other thing is that you clearly don't see anything close to atomic resolution. So they're all kind of low resolution structures. But still, during those days, there was still a big impact. So these are the UCT first. So this is associated with the electron microscope unit. So 1961 was the first EM virus images taken in Africa. It was using negative stain. And one of the things that hasn't been mentioned yet is that negative stain, which is kind of the old-fashioned low-resolution technique that a lot of people have kind of been mentioning, was actually invented by a South African. So this technique was invented by Sidney Brewer. And he has a great talk on YouTube about that. 1989, the first tomography reconstruction. That was Trevor and Mike Lawrence, who you heard earlier. 2003, the first negative stain reconstruction. And in 2022, I'll just skip ahead. The first close to 2 angstrom resolution cryo-EM reconstruction in Africa. That wasn't collected on our instruments. It was collected overseas at eBay. So there's a lot of research that's kind of been driven, both from within the EM unit as well as the users. So this is it's direct. You've just heard this particular talk. Teaching happens from within the unit. So one of the things that we really want to do is kind of train users. And one of the courses that's been run by us has recently been run through funding that we've received from Chan Zuckerberg, from the Chan Zuckerberg Initiative. We do outreach, so in other words, schools and various other peoples, including open days and this kind of thing, to try and promote the activities of the unit. Other projects have run from within the EM unit, so this was like an art project that was run. And really, here's the thing that I really want to talk about today. And the what I'm going to speak about today was one of the projects that runs through this program. So the idea behind this is that we really want to get as many people as possible. So all of the people that are sort of here today that might be interested in cryo-EM and others, we really want to get you to do cryo-EM. The crucial thing about this, and I think the difference with our training course compared to others, is that you have to have your own sample. And the sample has to be a good enough sample and it has to be appropriate. And so we are allowing people to come and basically paying for everything in terms of data collection and demonstrating to you how to reconstruct your sample. But you have to have a sample that is actually appropriate. And the reason behind this is because in many cases, people run training programs and a whole lot of students sign up and they kind of go through the whole thing and they, end up having a whole lot of fun, but they don't actually have a sample that they can use. And maybe 5 years down the line, they do, but they've forgotten everything. Maybe they never actually have a sample that's going to work. And so this is a really directed thing. In that, we are taking people through real experiments and we want to end up with real results. And that's really comes to the subject of this talk. Because most of these structures here, so not the angiotensin converting enzyme over here, and this was pre the training course, but it was really the kind of thing that actually led up to the training course. Is one of your pages. I'm sure that's what page is smiling. Each one of these structures over here was actually done by a participant of our training. Now so each one of these structures over here is really somebody's project and actually got some kind of insight out of this. And I'm just going to be focusing on 2 of the participants. So Ndile Zimba and Kefo Malebo. And they, between the 2 of them, they worked on this idea of an HIV vaccine. So they brought the sample to us. We had a look at it. We said, this sample is appropriate and you can be trained. What did they get out of this now? So the idea behind this whole thing, so an HIV vaccine, of course, it would be really great. You could reduce the incidence of HIV. There is no such thing as an HIV vaccine yet, and they've been very unsuccessful. And I just want to highlight some of the problems why an HIV vaccine has been unsuccessful. So the first thing is that there's extremely high genetic variability of HIV. In fact, a person who's infected with HIV, typically they have so many versions of the virus and so many mistakes inside them that they have one error for every point in the HIV genome. So in other words, that's an extremely high variability rate. So This allows HIV to then escape any kind of treatment that you're doing. Okay. Which then produces these escape mutants. Not only that, as these new variants keep coming up within an individual who's infected with HIV, you get this antigenic drift. So in other words, you keep having to produce antibodies for these new variants that are sort of arising within the person's body. So in other words, a successful vaccine needs to elicit an immune response that can target an extremely diverse HIV. There's another problem in that that the target of the HIV vaccine, which Kurt spoke about earlier, which is this HIV envelope protein, is covered in glycans. And so the protein itself is not accessible to antibody binding. There's another issue in that about 90% of the protein, this HIV envelope protein, which is the target, is misfolded. So these are some of the results that we got. We literally just put this HIV envelope protein on the grid. It has a preferential orientation, so it's always oriented down the threefold axis. And you just do a 2D classification. In other words, you're just looking for similarities between these particles, and it looks really, really horrible. And this is really typical because 90% of these proteins are actually incorrectly assembled. Not only are they incorrectly assembled, but they don't always assemble. So in other words, you get higher molecular weight, so they're not always trimers, and in some cases they're monomers. And there are the trimers. So even when you select the trimers, a lot of them are not correct. It's like correctly assembled. And this has what's called the decoy effect. So because they're all these different antigens, most of which, so 90% of which are actually inactive, the immune system kind of generates all of this sort of immune response to useless proteins, which then kind of means that you have a very broad sort of like immune response, and you can't generate neutralizing antibodies. There's another problem in that the virus itself has got a very low density of these proteins. So there they are over there, all these HIV envelope proteins. There are not so many of them. And so it doesn't elicit a very strong immune reaction. Okay. And the last issue is that if you just put in the sequence, so for instance, with the COVID-19 vaccines, you just have to put in this spike protein. You can either like inject the person with the spike protein or with mRNA, and you get this great response. With HIV, you get a lot of the immune response targeted to the base. So the base is not actually accessible, and so you get the biggest immune response to the part of the target that isn't actually accessible. So it's like a useless immune response. So that doesn't really work at all. Okay. So the idea behind this project was to try and get around every single one of these problems. Okay. Which is kind of tricky. So the first solution is to look at patients and find patients that are infected with a variant of HIV that's actually able to elicit a good, broadly neutralizing response. So that's these two over here. Okay. So the kinds of antibodies that are elicited by this particular variant is able to penetrate the glycan shield and actually produce an effective immune response. There are also a whole lot of things that are very well known in the field to try and stabilize this HIV envelope protein. So of course, the people that work in this field will know all of these. So that's like wild type HIV. That's kind of how it looks if you put it on the microscope. But if you do these various things, you can then stabilize it. And then your 2D classes look more like this. And the last thing that they wanted to do is attach it to a nanoparticle. So the idea behind this nanoparticle is quite important because if you attach the envelope protein to the nanoparticle, then you're going to conceal those epitopes at the base. So in other words, you're not going to have that kind of immune response that's going to just go nowhere. Not only that, you're also going to have so many antigens present that you're going to facilitate this linking to B cells, which is going to give you a more durable antibody response. But not only that, also the size of the whole particle is recognized by the immune system very much as a kind of a virus. And the immune system takes it super seriously. And so the effect of that is you get this drainage of them into the lymphatic system. And so in other words, in this particular size, which then gives you this really good immune response. And so Kimberly came along, she had this sample. She went through the training and collected these images over here, which when we classified, we could see that, yes, indeed, she had this nice particle. So this is the sort of underlying particle that she now wants to decorate with her HIV envelope proteins. And then when she added the HIV envelope proteins, we saw something kind of interesting, because you can see over here, there's a band. So in other words, this is a mouse. The HIV envelope protein is covalently attached to the nanoparticle protein. And so you get this increase in mass, but you only see a few of them that are actually attached together. You see an excess of HIV envelope protein, but yet, even though there's such an excess, you still have a lot of free nanoparticle protein. So in other words, you have like, I guess it's kind of difficult to judge the intensity, but you have a similar amount of nanoparticle that's not actually bound to the HIV envelope at all. So that's not doing anything in this whole thing. And when we look at particles, we can see HIV envelope attached to the nanoparticle, which looks quite cool, but the density is very, very low. And so this is just negative stain over here, but even in negative stain, we don't see that HIV envelope really being attached. We just see very, very weak density. And the result of that was that when this was injected into rabbits, I believe, I wasn't involved in that part, it was a very poor response. So in other words, the rabbits weren't producing neutralizing antibodies. I can go to the next one, maybe. There we go. Okay. Yes. Okay. So then we realized that this was no good. And the reason that it was no good was because the nanoparticle that was being used had a very, very small, actual protein size was quite small. And so there was an extreme symmetry mismatch between HIV envelope protein and the nanoparticle. And so it was very inefficient binding. And so Keiko then started her experiment. And you can see her nanoparticles look kind of similar. But when she decorated them with HIV envelope, we got this beautiful looking thing over here where you can see these HIV envelope proteins kind of coming off. And there is the 2D classification of hers. So you can see the particle there in the center, and even in negative stain, which is the super, super simple technique. So in other words, this is literally like a day's worth of work. You can stick this on, collect the images, and do your classification literally within a day. You can already see this really great result. So you can see that these particles are really binding nicely, and you have HIV envelope kind of attaching. But yet, we still saw the same effects. So you can see that over here, you can see HIV envelope attached to the nanoparticle. It's like the double band. It's the highest molecular weight. In the middle over here, you can see the HIV envelope protein, but you can still see the nanoparticle protein that is unbound. And we wanted to try and figure out what was going on over here. And so we did some higher resolution cryo-EM. Now I say higher resolution because this particular sample wasn't optimized. And so I think this is kind of an important thing to say, because in this particular case, we ended up collecting a lot of data. So we ended up collecting over 10,000 images, but we ended up with 1,400 actual particles. So it was very, very far from optimal. Even with a sample like that, you can see high resolution from the nanoparticle. So we got sort of 5.3 angstrom resolution, which is not amazing. There's definitely no record. It's far from the best reconstruction that we've done. But with only 1,400 particles, it wasn't too bad. We had a lot of heterogeneity with respect to the orientation of HIV envelope, but you can certainly see that it's HIV envelope. It's the correct size and you can kind of place it. So it's a kind of a good enough thing. And it's certainly good enough to answer the question of how is this particular thing binding? And why is there still nanoparticle that's unbound? And it's kind of difficult to see in this case over here, but if we sort of zoom in, we can see, so the nanoparticle is called Mi3, and Mi3 kind of sits over here. You can see there's 1, 2, 3 of them, and 1, 2, 3 of them, 1, 2, 3 of them, 1, 2, 3, surrounding, that's kind of surrounding this area over here, which is on the five-fold axis. So we can see there's like five-fold symmetry around here. And in the middle, we have HIV envelope, which is a trimer. And that trimer kind of sits in the middle. Because there are 5 possible places for it to bind, but only has 3 places that it can bind, you have this 5-to-3 ratio. So in other words, you're always going to have 2/3 unbound. And that's really the origin of that extra band. And so instead of when we first did this experiment over here, we thought, well, HIV envelope has got 3, it's a trimer. And this thing is icosahedral that we're binding it to, which means that it has 60 subunits. We're going to have 20 of these things binding, right? Because 20 times 3 is 60. But in fact, we didn't. We only had 12 because they were sitting on the five-fold axis. And that changes. So really, in prediction, you can kind of predict how this whole thing is going to assemble. But once it does actually assemble, you can see that it forms a different geometry, which means that it has a different antibody accessibility. So if you didn't know this, you wouldn't know exactly which of the epitopes were available for the antibodies to bind. Okay. And of course, a lot of people have been speaking about microscopes. And I just wanted to show this image over here. This is a kind of an old image. It's even worse now. These are high-end cryo microscopes around the world. And you can see that the US basically just looks gray. And Europe just looks gray. And certainly Japan and China are now gray. And India is becoming gray. And Australia is a little bit gray, I guess, but most of this is desert. But Africa doesn't have a single gray spot. So the whole of Africa doesn't have a single high-resolution cryo-electron microscope. And I think that's a real shame. And what we really want to do is say that the EM unit's position that we want to put it in is as some kind of a national facility. We really want to say that this facility, we have what we have, but if we can actually get a high resolution cryo-EM, we can position ourselves as a national cryo facility, and hopefully we can get large scale support from other researchers. So I quickly put together this acknowledgement slide. There's Claire van der Berg. She's actually running this training program. So I should have actually mentioned her earlier. Rose Chapman is the one who's kind of leading this HIV research, and that's why when I was describing, I didn't really know what was going on because it's not my work. It's Rose Chapman, and her 2 students are Pindile and where is Kefo? Kefo. Some of the other images that I showed are from Danielle Martin, who's over here in the audience, and she's being supervised by Tandeka, who's also here in the audience. Hello, hello. And they have some kind of association. I'm not sure exactly what it is, but so she'll definitely also be involved. She's the boss. Okay. Or is the boss. Manny was involved in some of the so those really horrible looking HIV envelope things because they were wild type. And Ed Ribicki is somehow involved with Rose Chapman. I'm not sure exactly how. And that's it.
Thank you very much, Jeremy. Don't run away. You may have to answer questions. Yeah. I hope people are going to interrogate Jeremy thoroughly. So,
Not about HIV, please.
about anything. But does somebody have a question for Jeremy? Yes.
The next training course.
It's happening all the time. So if you have a sample,
Yeah.
just contact. It's probably better to contact Claire.
Well, speak in the microphone.
Oh, sorry. Okay. Yes. Okay, so everybody can hear. So we're running the training program continuously, and if you have a sample, just contact us. I mean, in any case, even if we weren't running the training program, if you have a sample, just contact us, right? So we definitely want to try and promote this thing. The main issue with a sample, the main thing that cuts people off is the size of the protein that they're working with. And so we typically really only want to look at things that are above about 200 kilodaltons or so, just because it's very difficult for us to optimize the samples because the high resolution data collection is remote. And so we find that things tend to screw up. But of course, with Lizelle, it was quite successful. Yeah. She spent a long time. Yeah.
That was Topaz. We would never have done anything with the ACE without artificial intelligence. So
Yeah.
So it's entirely artificial intelligence thing, which by complete coincidence came out of the woodwork just as we were trying to pick our specimen, and we were absolutely failing.
Yeah. And that's it. It was relatively lucky because so the sample optimization, you'd normally need to see the sample while you're optimizing it. Now, of course, Lizelle couldn't really see it very clearly, and so we were sending a sample more or less blind. We kind of saw what we could, and then sent it overseas. But I mean, it could have easily been that the concentration was much too low or much too high. In that particular case, this is one of the problems with not actually having your own microscope, with having to always send it off. Yeah.
And no way of recognizing the protein molecules.
Yeah, for sure. So it worked in the end, but we definitely don't want to do that, yeah. So if you have large samples, viruses especially.
Okay, next training is more important to Tuesday.
Thanks, Claire.
Pete, what you said.
Oh, so Claire just said that the next training session is from the 14th to the 26th of October. Yeah. So even though we're getting samples in continuously, we now are trying to train more than one person at the same time, because previously it was all one-on-one. So it's very difficult to get done. Yeah. Good.
Can I just ask a question? Sorry, Trevor.
Go ahead. Come.
Yeah, no, it's fine. I just wanted to know. So,
people, are you talking to Mike?
Yeah. So just based on what you said in your last talk, I just wanted to from a technical perspective. You have a nanoparticle. You can have different antigens on it, more than one type, and you can look and you can compare the orientation of or how these different antigens are situated on a nanoparticle, and it can be more than one type of antigen on it.
So that. Yeah. So in this case, there was only one antigen on, but it is possible to split those up because that just represents some kind of heterogeneity. So in this case, the heterogeneity that we had was relatively complicated because HIV envelope protein that was sitting on the nanoparticle actually was quite flexible, and so it can kind of move around like this. Because we didn't have that many particles, we only had 1,400. We couldn't really do sufficient 3D classification to see high resolution for every single one of those HIV envelope proteins. Part of the reason that we had that limitation is because there was a symmetry mismatch. And so the icosahedral symmetry is really nice, but we couldn't impose icosahedral symmetry because the threefold symmetric HIV envelope is binding on the fivefold axis of the icosahedron. But if you have sufficient number of particles, then you don't have to use that symmetry as this powerful constraint. And so what you can do is you can just reconstruct the nanoparticle, and then you can say, okay, well, I'm not going to mask out each of the different locations where you could have this different antigen. And then you can visualize each of the antigens separately by classifying that particularly. So by using 3D classification. Now those techniques obviously add an extra degree of sort of complication, but they do provide very important information, especially if you want to have several different antigens on this nanoparticle, and you want to make sure that it's definitely heterogeneous and it's well mixed, and you have a certain ratio of antigens, then you can get that kind of information. Yeah.
Thanks, Jeremy. We now have a very short 10 minutes